Transcription occurs within the nucleus and is the process by which mRNA, or messenger RNA, is formed. The 3' to 5' strand of DNA, called the antisense strand, is unzipped by the RNA polymerase, detaching it from the 5' to 3' strand, called the sense strand. Only a small section is unzipped, containing the gene to be transcribed. The first stage is initiation. The binding of the RNA polymerase to the promoter initiates the transcription of a gene. In elongation, the RNA polymerase uses free-floating RNA nucleotides to form a 5' to 3' strand complementary to the antisense strand. The resulting mRNA strand is identical to the sense strand, with the exception that thymine is replaced by uracil in the sequence. The mRNA is detached from the DNA strand in termination, the ending marked by a sequence known as the terminator. Then, in post-transcriptional processing, the introns, RNA segments that don't code for proteins, are spliced out, and the remaining segments are called exons. The mRNA then passes through the nuclear membrane into the cytoplasm, where translation occurs. The translation process starts at the 5' end and ends at the 3' end. A segment of 3 bases on the mRNA is called a codon, and each codon codes for one amino acid. The 4 steps of translation are initiation, elongation, translocation, and termination. Initiation starts when the smaller subunit of a ribosome encounters the mRNA and attaches itself. It scans for a starting codon, and when it finds one, the bigger subunit joins it to form a complete ribosome. The ribosome's structure consists of these 2 subunits, the 40S and the 60S, both working together in translation to string the amino acids together, elongating the polypeptide chain. It is made of protein and ribosomal RNA. The ribosome contains the catalytic center responsible for forming peptide bonds, the bonds between the amino acids that form a chain. tRNA is activated when an amino acid bonds to the tip. Amino acids bond with different tRNA based on their anticodons. Elongation occurs when the complementary mRNA codon matches the tRNA anticodon and releases the attached amino acid to the polypeptide chain. The complementary pairing of codon and anticodon move through the ribosome in the 5' to 3' direction, allowing the next codon and anticodon to enter the decoding center of the ribosome. This is known as translocation. The polypeptide chain is terminated and released once the ribosome reaches a stop codon, UGA, UAG, or UAA. These are the only 3 codons that don't code for an amino acid. The ribosome's subunits dissociate and it separates from the mRNA. Once fully formed, the polypeptide chain wraps on itself or joins with other polypeptide chains to create proteins.